Synthetic pyrethroids. Synthetic pyrethroids are synthesized derivatives of naturally occurring pyrethroid, which are taken from pyrethrum, the oleoresin extract of dried chrysanthemum flowers. The insecticidal properties of pyrethrums are derived from keto-alcoholic esters of chrysanthemic and pyrethroic acids. These acids are strongly lipophilic and rapidly penetrate many insects and paralyze their nerve system. Mode of action: pyrethroids are axonic excitotoxins, and toxic effects of which are mediated through preventing the closure of the voltage-gated sodium channels in the axonal membranes. The sodium channel is a membrane protein with a hydrophilic interior. This interior is shaped precisely to allow sodium ions to pass through the membrane, either the axon, and propagate an action potential. When the toxin keeps the channels in their open state, the nerves cannot repolarize, leaving the axonal membrane permanently depolarized, thereby paralyzing the organism. Pyrethroids can be combined with the synergist pipernoyl butoxide, a non-inhibitor of key microsomal cytochrome P450 enzymes, from metabolizing the pyrethroid, which increases its efficiency, lethality. It is likely that there are other mechanisms of intoxication also. Disruption of neuroendocrine activity is thought to contribute to their irreversible effects on insects, which indicate a pyrethroid action on voltage-gated calcium channels and perhaps other voltage-gated channels more widely. Alethrin. Alethrin was the first synthetic analog of pyrethrum developed in 1949 by Green and LaForage. Alethrin is more common name for rs3-allyl 2-methyl 4-oxocyclopentane 2-enyl IRS cis-trans chrysanthemate. It was first described by some scientists and introduced by Sumitomo Chemical Company. It has contact, stomach, and respiratory action and brings about quick knockdown of flies and mosquitoes when applied in combination with synergists like pipernoyl butoxide. The acute oral LD50 for rat 585 to 1100 mg per kg, dermal for rat greater than 200 less than 200 mg per kg. Its registered formulations are 0.5% coil, 2% and 4% matte, 0.5% aerosol, and 3.6% liquid. Then pyrethrin. The pyrethrins are powerful contact insecticides that appear to be poor stomach poisons. A characteristic action of pyrethroid is the rapid paralysis or knockdown effect and substantial recovery that follows it. Its recovery is due to rapid enzymatic detoxification in the insect to bring about a mortality equivalent to the knockdown effect. About threefold increase in dosage may be required. Compounds such as piperoyl butoxide, propyl isome, and sulfoxide are known to inhibit the detoxification enzymes and increase the toxicity of pyrethroid. These synergists are used at 10 parts to 100 parts of pyrethroid. LD50 for rat, oral 200, dermal for rat 1800. Pyrocan E222, 1 part of pyrethrin plus 10 parts of piperonyl butoxide is used for the control of coconut red palm beetle. Then pyrocolloid, a colloidal form at 1 in 400 to 600 parts of water, is useful against aileroided white flies and scales in household sprays and as a repellent against external parasites of livestock. Pyrethrum is useful. It is also mixed with stored grains for control of stored grain pests. Inorganic compounds as insecticides. Inorganic insecticides comprises of compounds of mineral origin and elemental sulfur. The following are some groups of inorganic compounds which find use as insecticides, rodenticides, and molluscicides. Arsenic compounds. In an arsenical compound, inorganic compounds as insecticides. Inorganic insecticides comprises of compounds of mineral origin and elemental sulfur. The following are some groups of inorganic compounds which find use as insecticides, rodenticides, and molluscicides. Arsenic compounds. In an arsenical compound, the total arsenic content present and the proportion of water-soluble arsenic are of considerable importance. If a compound has a very high water-insoluble arsenic content, it is otherwise readily soluble in insects' digestive juices. It is considered to be ideal for use as an insecticide, as water solubility of the arsenic may result in entering the foliage and causing burning injury to plants. Water insoluble compounds are preferred for insect control. The arsenics are generally more stable and safer for application on plants than the arsenides, and the latter are therefore used mainly in poison baits and not on insects, or not on plants, as they are phytotoxic. However, the arsenides are less toxic to insects than the arsenides. In insect, arsenides cause regurgitation, torpor or sluggishness, and quiescence. Disintegration of the epithelial cells of the midgut with vacuolized cytoplasm and clumping of the chromatin of the nuclei are the effects noticed in poisoned insects. Slow decrease in oxygen consumption is also evident, and kill of the insects is primarily due to inhibition of respiratory enzymes. In plants, foliage injury due to water soluble arsenicals appear as a wilting followed by a browning and shriveling of the tissue, and also with interveinal necrosis. To reduce this effect, equal quantity of lime is added. Then lead arsenate. It was first used as an insecticide in 1892 by Moulton for the control of gypsy moth. The acidic orthoarsenate and the basic orthoarsenate were the forms used in insecticides, and the former predominated in commercial product. It is a stomach poison with little contact action. It is not in large scale use now. Calcium arsenate. Lead arsenate, calcium arsenate. It was first used by about 1906 as an insecticide. A commercial product consisted of tricalcium arsenate and acid calcium arsenate, and the former predominated. Calcium carbonate and lime are used in excess. In a freely manufactured product, water soluble arsenic is found in only small percentage. When stored, water soluble arsenic get liberated due to decomposition by carbon monoxide and ammonia in the atmosphere and dilute alkaline solutions. Therefore, the product, if it is more than a year old, should not be used. It is a white flocculent powder and formulated usually as a dust containing a pink colored matter and 25 to 30% metallic arsenic equivalent. The dosage calcium arsenate at 0.675 to 1.350 kg with equal quantity of slacked lime in 450 liters of water. LD50 for mammals. Oral ranges from 3.35 to 100. Being a touch poison, it was mainly used for the control of leaf-eating insects. Then white arsenic, also known as arsenic trioxide or arsenic oxide. It contains 75% arsenic and is used in poison baits for rats, grasshopper, armyworms, ants, and cockroaches. LD50 for rat, oral 20 to 300. Sodium arsenate. It is a white to yellowish grayish powder. It is used for baiting ants and locusts and for stock dip treatment for control of ticks or weed control. LD50 for mammals, oral 10 to 50. Then Paris green, it is a double salt of copper acetate and copper arsenate. Fluorine compounds. Fluorine compounds. Since 1890, fluorine compounds have found use as insecticides. They are principally stomach poisons and to a limited extent contact poisons. The kill is rather more rapid than fluorine compounds, than which arsenicals. Their insecticidal properties are related to the fluorine content and solubility in the digestive juices of the insect. Phytotoxicity of the compound is attributed to its higher water solubility. Fluoride poisoning produces spasms, excessive muscular contraction, and sudden convulsive movement, regurgitation, flaccid paralysis, and death. In insects, metal containing enzymes such as enolase require magnesium for making up deficiencies, and in poisoned insects, fluoride with magnesium forms a complex magnesium fluorophosphate and thus inhibits phosphate transfer in oxidative metabolism. Then sodium fluoride. It is a white powder, but the commercial product of 93 to 99% purity was colored. It was used in baits for the control of cockroaches, earwigs, cutworms, and grasshopper and chewing lice on animals and poultry. LT50 for rat, oral 200. It is highly phytotoxic. Then sodium fluoroaluminate, also known as sodium aluminum fluoride or sodium aluminum fluoride, aluminofluoride. Markovich and Stanley used it first as an insecticide in 1929. It is found up to 98% in naturally occurring mineral cryolite, but can also be manufactured synthetically. It was used for the control of chewing insects. LD50 for rat, oral 13.5. Sodium fluorosilicate. It is phytotoxic but a soluble form finds use in mouth-proofing fabrics. LD50 for rabbit. Then sulfur compounds. Sulfur, it is primarily a fungicide and acaricide but may serve as a diluent for some insecticidal dust. It is usually formulated as a fine dust, 95% of it passing through a 325 mesh sieve with up to 10% inert material to prevent beading. The flow of dust is also made more free by addition of 3% tricalcium phosphate. It is also formulated as a wettable powder. Effectiveness increases with fitness of sulfur particles. It is generally non-toxic but irritating to eyes. Then lime sulfur. It is the aqueous solution of calcium polysulfide. It was first used as a fungicide in 1852 by Grisson and later in 1886 by Dussey for the control of Sanjo's scale. It is prepared by sulfur solution in calcium hydroxide suspensions, preferably under pressure in the absence of air. Calcium pentasulfide and calcium tetrasulfide found in the mixture are the active materials of insecticidal value. Scales, mites, and aphids, and fruit on fruit trees besides powdery mildew are controlled by the preparation. Then Bordeaux mixture. A combination of copper sulfate, lime, and water, is an effective fungicide and bactericide, and that has been used for decades to control decreases of fruit and nut trees, vine fruits, and ornamental plants. These natural minerals, when mixed in the correct order, provide long-lasting protection to plants against diseases. In preparing it, the CuSO4 and the lime are dissolved separately in water and then mixed. Calcium oxide, burnt lime, and calcium hydroxide, slacked lime, gave the same end result since an excess of water is used in the preparation. Bordeaux mixture is very effective in vineyards, fruit farms, and gardens to prevent infestation of downy mildew, powdery mildew, and other fungi. Bordeaux mixture achieves its effect by means of the copper ions of the mixture. These ions affect enzymes in the fungal spores in such a way as to prevent germination. This means Bordeaux mixture must be used preventatively before the fungal disease has struck. The ability of Bordeaux mixture to adhere to plants in rainy weather makes it an excellent choice for a winter fungicide. Applying Bordeaux after trees break dormancy, generally it's recommended because it can injure leaves. Next topic, control of microorganisms by physical and chemical agents. Sorry, that's not related in entomology, sorry. Next topic, botanical insecticides. Toxicants derived from plants are used in insect control, and among them nicotine, pyrethrum, rotenone, etc. are well known. Certain plant products also find use as nematicides, then insect attractants and repellents, and as diluents in insecticidal formulations. Nicotinoids. nicotine 131 methyl pyridyl pyridine. The principal alkaloid in tobacco was discovered in 1828, though tobacco itself was used as early as 1763 in insect control. Nicotine is found in the leaves of Nicotiana tabacum and Nicotiana rustica from 2 to 14%, also in Duboisia hopewoodi and Asclepias syrica. Among the 12 alkaloids present in tobacco, nicotine is most important one, contributing about 97% of the other two of insecticide value are nornicotine and anabasine. Nornicotine occurs in 1 to 2% in shoots of Anabasis aphylla and about 1% in N. glauca. The alkaloids are appreciably volatile and basic, whereas nicotine sulfate is more stable and less volatile. It is a nerve poison, being highly toxic when absorbed through the cuticle or taken in through the trachea or when ingested. Nicotine and anabasine affect the ganglion, blocking conduction at higher levels. Nicotine sulfate containing 40% alkaloids is safer, more convenient to use, and free alkaloids is liberated by an addition of soap, lime, or ammonium hydroxide to the spray solution. It was used for the control of cardamom thrips. Dust formation of nicotine sulfate released nicotine in the presence of moisture. It also finds use in aerosol. Tobacco decoction useful for controlling aphids, thrips, etc. can be prepared as follows. Boil 1 kilogram of tobacco waste in 10 liters of water for 30 minutes or steep it in cold water for a day. Then make up the infusion for 30 liters and add about 90 gram of soap. Addition of soap improve wetting, spreading and killing properties. Nicotine does not leave any harmful residues on treated surface. LSD for LD50 for rat. I'm sorry, not LSD. LD50 for rat, oral 50 to 60. Pyrethroids. Pyrethrum as an insecticide was first used by about 18,000 in Transcaucasus region of Asia. Initial plant sources of pyrethrum were Chrysanthemum coccinum and C. carneum compositae. However, only since 1840 other Chrysanthemum cinereifolium became the chief source. The insecticidal properties of pyrethroids are attributed to 5 esters: pyrethrins 1, 2 and 3, cinerins 1 and 2 and jasmolene 2, which are predominantly found in the akins of flowers from 0.7 to as much as 3%. The esters are derived from two acids, chrysanthemic acid and pyrethric acid, and alcohols cinerolone and pyrethrolone and jasmolone. The esters containing pyrethron are called pyrethrin 1 if the acid is chrysanthemic acid, the pyrethrin 2 if pyrethric acid. Similarly, those containing cinerolone are called cinerolene 1 if the acid is chrysanthemic acid, the cinerolene 2 if pyrethric acid. Pyrethrum powder is prepared by grinding the flowers. The powders mixed with a diluent such as talc or clay is known as pyrethrum dust. It is prepared just before use as otherwise it gets deteriorized rapidly. It is also used as emulsion, solution and aerosol. Pyrethrum is unstable in air, light, moisture and alkali. The residues deteriorate very rapidly after application. The pyrethrins are powerful contact insecticides but appear to be poor stomach poisons. A characteristic action of pyrethroid is the rapid paralysis or knockdown effect and substantial recovery that follows it. This recovery is due to rapid enzymatic detoxification in the insect. To bring about the mortality equivalent to the knockdown effect, about threefold increase in dosage may be required. Compounds such as piperoyl butoxide, propyl isobe, and sulfoxide are known to inhibit the detoxification enzymes and incorporate the toxicity of pyrethroid. These synergists are used at 10 parts to 100 parts of pyrethroid. LD50 for rat, oral 200, dermal for rat 1800. Then pyrocone E is used for the control of coconut red palm weevil. Pyrocolloid, a colloidal foam at 1 in 400 to 600 parts of water, is useful against whiteflies and scales. In household sprays and as a repellent against external parasites of livestock, pyrethrum is useful. It is also mixed with stored grains for control of stored grain pests. Rotenoids. Rotenone is the main insecticidal compound of roots of Derris and Longocarpus. Rotenoids, main insecticidal compound of roots of Derris. Derris root is a well-known fish poison. It was first used in 1848 against leaf-feeding caterpillars. However, rotenone was isolated in 1859 by Geoffrey and Jeffrey. Through rotenone is reported from 68 species of leguminous plants. Principal commercial sources are Derris elliptica. Among the toxicants in roots and seeds, rotenone is the chief compound occurring in Derris roots, and the others being elliptone, sumatrol, malacol, toxicrol, and deguilin. It is oxidized non-insecticidal compound in the presence of light and air, and hence rotenone residues are difficult to find after 5 to 10 days in normal sunlight. Insects poisoned with rotenone show a steady decline in oxygen consumption followed by paralysis and death. It is very specific, being highly toxic to fish and most insect species but almost harmless to warm-blooded animals except pigs. Neem, Asa Directa indica. Kernels of neem tree possess extraordinary gustatory repellent properties which are attributed to the active principles nimbidin T, meliartriol, and azadirachtin. Neem seed kernel powder mixed at 1-2 parts with 100 parts of wheat seed affords protection against stored product pests for 9 months. Neem cake extract. 1 kilogram of neem cake is soaked in 5-liter water for a week with frequent stirring. Filtered through the cloth and made up of 100 liters extract. Sprayed on citrus minimizes leaf minor incidents. Azadirachtin. Azadirachtin, it is extracted from the kernels of neem tree and yellow-green powder with a strong garlic sulfur-like odor. Agonisting the insect hormone ecdysone, it is also an antifeedant. Its acute oral LD50 for rat is shown. Neem extract concentrate generally contains azadirachtin and formulated as emulsifiable concentrate. Azadirachtin. Nimbicidine. It is yet another alkaloid of neem product introduced for pest control in India. Nimbicidine. Nicotinoids, pyrethroids, retinoids, and neem. Azadirachtin, nimbecidine. Next, agricultural entomology, pests of cardamom. In India, more than 50 species of pests are known to attack cardamom. Some of them are cardamom thrips, rhizome borer, cardamom whitefly, hairy caterpillars. Cardamom thrips. Sciothripes cardamomi, Rhizoroptera thripidae, nymph and adult, commonly called the cardamom thrips. Most harmful pest in South India, first discovered in 1933 in an estate in Velparai on Anamala Hills. Kidney-shaped eggs. Transparent larvae, approximately 1 by 25 inch in length. light brown in color. Pupae resemble the larvae except for wing pads, incompletely developed wings. Adult is minute, dark grayish brown with fringed wings. Sap feeding insects. The head and abdomen of the strips are dark gray brown. Thorax and legs are pale yellowish brown. Females measure approximately 1 by 4 inch in length. Males are slightly smaller. Larvae and adult inhibit the inflorescence. Seeds, tender leaves, swollen leaf bases, and the aerial cardamom plant in large group. Life cycle biology. Life cycle involves direct and hemimetabolous metamorphosis. Direct and hemimetabolous metamorphosis. The life cycle egg to adult is 25 to 30 days. Reproduction is mostly sexual. True virgin females are able to lay viable eggs during the summer. The white kidney-shaped eggs are inserted in leaf sheaths or flower brackets by the sharp ovipositor of the adult. Eggs hatch in about a week. Larvae. The larval stage consists of 3 larval stages separated by moths in the course of 50 to 21 days. Hemimetabolous metamorphosis. The life cycle egg to adult is 25 to 30 days. Reproduction is mostly sexual, although virgin females are able to lay viable eggs during the summer. The white kidney-shaped eggs are inserted in leaf sheaths or flower bracts by the sharp ovipositor of the adult. Eggs hatch in about a week. Larvae. The larval stage consists of 3 larval stages separated by molts in the course of 50 to 21 days. Pupae. There are 2 pupal stages, the pre-pupa and pupa, neither of which are feeding stages. Pupae becomes adults in 10 to 15 days. Adults females lay as many as 70 eggs in different feeding parts of the host plant. The head and abdomen of the strips are dark grey brown. High temperature and low humidity favors the growth of this insect. Analyze strip life cycle. Egg. First instar larva, second instar larva, propupa, pupa, adult egg, pre-pupa. Damage. Both the nymph and adults are harmful. They lacerate the tissue and suck plant sap from blossoms, spores, and other parts. The injured tissue form a corky layer on the capsule surface, which appear as scales. Such capsules appear stunted, malformed, and shriveled with gaping slits on the skin. The condition is popularly known as cardamom itch. Seeds from infected capsules give poor germination. At panicle formation stage, infestation causes stunting of panicles and shedding of flowers and capsules. Scraping of capsules lower their quality. Such pods lack their fine aroma and the seeds within are also poorly developed. Then control measures. Non-chemical control. Natural enemies of cardamom trips. Predators, lacewing, big-eyed bug. General cultural practices involve sanitation such as removal of alternative hosts and plant debris within and outside the field. Regulation of shade in open areas. Remove any dry leaves, leaf sheath and old panicles prior to chemical spraying results in the reduction of strip population by 1/3. Chemical control. Testing with quinalphos 1.5% once in a month. Spraying of phosalone, quinalphos and profenphos during the flowering and seeding season are found effective. Quinalphos and profenphos. Cardamom is cross-pollinated mainly by honeybees, so it is desirable to use an insecticide which is non-toxic to honeybees. Cardamom hairy caterpillar, scientific name Euparocardamome, Leptoptera bombicidae caterpillar, South India. Eggs dome-shaped, lay on undersurface of leaves of shade trees. Dome-shaped leons, undersurface of leaves of shade trees. Larvae is hairy, robust brush black with pale brown head, white hairs and dorsal conical tuft of hairs. Pupation take place in soil. Adult large mouth, pale yellow with wavy lines and a series of spots near the outer margin of wings. Life cycle indirect development, hemimetamorphosis. Adult female moth lay eggs about 500 in undersurface of leaves of the tall shades of trees of cardamom plants. So egg hatch out to hairy larvae within 15 days. They feed on the tender leaves of shade trees using silken threads which they secrete or jump down to cardamom plants. Larvae undergoes 10 instars in 5 months. When full growth is attained, they came to soil and pupate in underground soil at a depth of 2 to 2.5 inch.